Good morning, everybody. My name is Rhonda Henshaw-Powell. I'll be your moderator for today. Just a couple of housekeeping requests before we get started. Please remain on mute. This can be particularly distracting for both of our speakers today and for the audience as well. Also, we have a series of panelists available online, some of our ACD support and PAS experts, waiting in the wings to answer any questions that you have during the presentation. So as you're listening to today's speakers, please do pop your questions in the questions box and our team will be addressing them as we go through. We have two excellent speakers today, Shauna Levinson and Alpana Kumari, both members of the PAS and support teams, and they're going to be presenting the clinical assay development process using ACD's ISH assay services. So with that, I'd like to hand over to our first speaker, Shauna Levinson. Thank you very much.
Hello, everybody. Hope you are all staying safe and healthy. We are excited to have you here today and present to you about our expertise in RNA-ISH and how this can be applicable to your clinical assay development needs. Again, my name is Shauna Levinson, and I am a Senior Research Associate here at ACD and part of the Pharma Assay Services team. I will be going over the RNAscope technology as well as the CDX development and clinical assay workflow. I will then hand it off to my colleague, Dr. Alpana Kumari, to go over examples of RNAscope use in the clinic. I'd like to start with a snapshot of Bio-Techne as a whole. Bio-Techne combines a wide variety of brands, companies, and solutions that are available to help with your research. So what is RNAscope and how is it relevant to spatial transcriptomics? Traditional transcriptomic analyses have relied on bulk tissue analysis, such as PCR, which provide an average level of gene expression in a sample, but loses the spatial and single cell information. This is akin to a fruit smoothie in which you can only guess at what fruit it contains. The advent of single cell transcriptomic analyses such as single cell RNA sequencing have now been able to provide molecular information at the single cell level, and to come back to the fruit analogy, can now provide you the menu and tell you what fruit and how much of each fruit went into the smoothie. However, these techniques still don't provide information on how those cells are organized in the tissue context. This is where spatial analyses come into play. Single cell multiplexing spatial analyses that detect RNA can provide transcriptomic information with spatial organization while retaining single cell resolution. So now, instead of an average blended smoothie with mystery ingredients, you can see all the fruit and how it is spatially arranged. The growth and adoption of RNAscope technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then, there have been over 2,700 peer-reviewed papers published using the RNAscope technology in numerous journals, including many top-tier ones. The RNAscope technology is highly relevant across multiple fields of research, with over 40% of the publications in cancer research, followed by neuroscience and infectious diseases. Furthermore, the incorporation of the RNAscope technology into single-cell RNA sequencing workflows is demonstrated through the rapid growth of publications using the assay to confirm and spatially map single-cell RNA sequencing data in the tissue context, with over 80% of these single-cell papers in high-impact journals, including Nature, Cell, and Science, as well as top journals in specialty areas such as pathology, oncology, and neurology. The RNAscope technology is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single cell level. It consists of 3 parts. a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal to noise ratio, and lastly, visualization of single RNA molecules as dots. The assay allows for spatial mapping of mRNA, long non-coding RNA, splice variants, point mutations, and highly homologous and short sequences in cells and intact tissues, of which can be visualized with either fluorescent or chromogenic labels. The assays can be performed on a wide variety of sample types, including FFPE tissues, fresh frozen or fixed frozen tissues, PBMCs, and culture cells. Diving slightly deeper into how the technology works, the first key feature of the RNAscope technology is the probe design. We depict the oligonucleotide target-specific probes as Zs to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z complements and hybridizes to the target transcript. The top of the Z is the base for the amplification structure. When the two Zs in a ZZ pair hybridize in tandem to the target sequence, it creates a unique binding site upon which a preamplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of 1,000 bases or more will consist of 20 ZZ pairs pulled together and designed to hybridize in tandem along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind to the target RNA sequence in order to generate enough signal for molecular detection. Here we walk through how signal is amplified with the RNAscope assay. The two key features of our technology are the probe design, as we just described, and the signal amplification system. Once a ZZ pair is bound in tandem to the target, a preamplifier can bind the opposite end of the pair, after which the amplifiers bind to the preamplifiers, followed by which the chromogenically or fluorescently labeled oligos bind to the tree. This amplification system allows for visualization of single RNA transcripts, and hence very low levels of target can be detected. On the other hand, if only one Z of a pair binds non-specifically to a sequence, the preamplifier will not be able to hybridize and the amplification tree will not form. As a result, no signal will be detected. The probe design and amplification system at Strategies provide for high sensitivity and specificity of detection. Single molecule detection is what we focus on and what is possible with the RNAscope assay. This is illustrated in an experiment that was published in the first publication demonstrating the RNAscope assay. One plate of Hela cell culture was run using the RNAscope assay probe for HER2, and the other was lysed, and then the solution was probed for HER2. With the RNAscope experiment on the left, you can count HER2 mRNA as the green fluorescent dots, which averages about 14 dots per cell, or 14 HER2 RNA copies per cell. On the right, a standard curve was used to determine the average HER2 RNA copies per cell in the lysate, which averaged about 17 copies per cell. So the two assays give very similar RNA copy readouts. The point being here that when we see a dot using RNAscope, it translates to a molecule of transcript. Here is an example of RNAscope being used to tease out heterogeneity in gene expression within the tumor. This is a duplex chromogenic staining for PD-L1 and LAG3, which are two common targets in immuno-oncology. It was found that in this tumor, PD-L1 is primarily expressed in the tumor compartment, whereas LAG3 is primarily expressed in the stromal compartment. This information is lost in the whole core analysis. The RNAscope technology includes 3 unique assays to detect RNA in the tissue context at single molecule sensitivity with single cell resolution. This means that a single dot represents a single RNA molecule or target. These 3 assays are RNAscope, BaseScope, and microRNAscope. The RNAscope assay is applicable for mRNA or non-coding RNA greater than 300 nucleotides in length and is available in singleplex or duplex chromogenic or as multiplex fluorescent assay, which can detect up to 4 targets simultaneously. BaseScope is detected for it's designed for the detection of exon junctions, splice variants, point mutations, and highly homologous or short sequences between 50 and 300 nucleotides in length. BaseScope is available as a singleplex or duplex chromogenic assay. MicroRNAscope is designed for the detection of antisense oligos, microRNAs, siRNAs, and other RNA sequences between 17 and 50 nucleotides in length, and is available in chromogenic. Pardon. Now I'd like to give you a brief overview of ACD's Pharma Assay Services. PAS is a team of 20 plus highly trained specialists, scientists, and pathologists that are experts in RNAscope performance and analysis that will ensure delivery of the best data possible. We provide services for 20 of the top 20 pharma companies in the world. In general, we deliver greater than 15,000 stain slides per year to customers in more than 20 countries. As shown here, we can help with acquisition of tissue samples through our trusted chain of tissue providers and qualification of these for the RNAscope-ish assays. We also work with third-party CROs and can easily manage the embedding and sectioning of the samples in your studies. We perform the RNAscope, BaseScope, and microRNAscope assays, which are routinely provided through our service panel, provide quantitative assessment, and put all this together into a report, which includes our findings as well as the materials and methods of the study. We also wanted to share with you something about how we manage quality systems and security for the data generated through PAS. SOPs govern the PAS workflow, including staining procedures, sample tracking, data management, and data QC. Each assay run includes Hela cell technical controls. The Hela positive and negative control slides must pass strict QC requirements in every assay run. Any runs that fail the run suitability requirements will be repeated. The data that we generate here is shared through a secure enterprise file sharing system called Ignite. Access to this data is highly restricted. The Pharma Assay Services team can help you with drug discovery through the phases, from target discovery to development. We can help with target screening, efficacy screening, DMPK studies, safety and toxicology studies, as well as performing early feasibility assessment for development in phase 1 clinical trials. Here we have PAS, the PAS CDX and clinical assay development workflow. From assay development and precision testing, the pathologist-guided scoring and analysis, the written SOP, and reporting to assay transfer and cross-validation. This slide illustrates a SOW or analytical plan and assay development. Here on the left, you see a Pharma Assay Services SOW template. Assay development would include choosing an assay platform. It might include optimization steps for sample pre-treatment and probe optimization and acceptance criteria. Our validation parameters include specificity and sensitivity, repeatability and precision, dynamic range, robustness and stability. Assay precision testing might include inter-instrument and inter-day reproducibility, as well as same-day repeatability. PAS also uses pathologist-guided scoring criteria for sample analysis. As seen here, a pathologist has appropriately annotated the different tumor and tumor-associated stromal compartments for the identification of heterogeneous expression patterns. Histopathology notes can also include things like tumor load and visual H scoring. A pathologist is also available to discuss and review data. We also wanted to share with you how our lab manager and project manager work very closely with the clinical specimen collection site, as well as the central data management lab to work through data transfer specifications and agreements. Our project managers meet on a regular basis with the sponsors and data management sites for smooth project and data flow. Lastly, as you see at the bottom there, we have a streamlined process for batch processing of samples as they come in, and we work with our sponsors to provide these to report data back with minimal timelines. This slide demonstrates an example of a batch workflow. For 1 to 10 samples per batch, and depending on the assay, on day 1 we would receive the samples and begin QC staining. Day 2 would include post-processing and review of the QC slides. Day 3 would include marker staining, and day 4 post-processing and slide scanning. On day 5 there would be slide scanning, scoring, and analysis, and day 6 would include reporting data QC and project delivery. PAS utilizes LabVantage, which is a widely used LIMS in the industry for auditable and efficient sample tracking throughout studies per GCLP guidelines and to ensure data sample security. The Pharma Assay Services Group is here to provide you with data and to perform any of the RNAscope assays for you. The PAS group has profound expertise with over 15,000 slides delivered per year, access to 14 automated staining systems from Leica and Ventana, digital slide scanners for both bright field and fluorescence images, Hela software for quantitative image analysis, board-certified pathologists are on staff, and a tissue bank with qualified human normal and tumor tissues available for immediate screening. Once your study is complete, you will receive a report containing representative images, materials and methods, summarized data analysis, and the study summary and conclusions. You will also receive full slide digital scans, the stained slides, and any unused material. ACD has established protocols for clinical assay transfer to CLIA certified labs for prospective use. And validation, cross-validation. We provide 3-way conference calls between ACD and the sponsor and the CLIA lab to go over things such as assay and protocol transfer and verification and cross-validation and pathologist scoring criteria. We can also provide an on-site visit with one of our field application scientists to assist in the transfer process. And now I will hand over the presentation to Dr. Alpana Kumari to go over examples of RNAscope use in the clinic.
Hello everyone, can you hear me?
Yes, we can. Thank you, Alpana.
Hi, my name is Alpana Kumari, and I am a member of Pharma Assay Services at ACD. And in the next few slides, I will show a few case studies illustrating that RNAscope has been proven to be effective means of detecting a biomarker that would be predictive of patient response to cancer therapeutics. First of all, I would like to shed some light on the features of RNAscope technology that makes it suitable for diagnostic tests. RNAscope works best with FFPE tissues, which can be fresh or archival tissues. In addition to FFPE, RNAscope works equally well with frozen samples, and the flexibility in the sample type is certainly helpful for clinical studies. The RNA expression data can be visualized with morphological context, and its sensitivity is at the level of single RNA molecule, which is much higher than IHC. The data analysis also allows flexibility, as the scoring can be done visually by a pathologist, or it can be quantitatively done using a software. Finally, it is performed on automated instruments ensuring that it will be reproducible between batches. This is a chart comparing RNAscope with other available assays. And as shown here, RNAscope is the only platform that provides most sensitive and specific data in morphological context. And it is flexible in terms of tissue requirements, as mentioned earlier. It can be performed on almost any types of sample, be it FFPE, fresh frozen, fixed frozen tissues, or cell lines. Furthermore, specific RNAscope probes can be designed for any target or biomarker. It can be performed as a single plex using one probe at a time, or it can be multiplexed to visualize multiple targets in specific cell types. And finally, the quantitative data analysis will provide cell-by-cell data with single molecule sensitivity. This is a piece of data from clinical cancer research published in 2014, showing Col11A1 protein and RNA expression in primary, metastatic, and recurrent serous ovarian cancer tissue. The authors saw good correlation between protein and RNA during cancer progression. RNAscope showed better dynamic range of expression than IHC, and RNA provided higher resolution than protein. As we can see here, the top panel is showing IHC and the bottom panel is RNAscope, and it is clearly visible that RNAscope is showing a better dynamic range as compared to IHC. Therefore, they determined that RNA serves as a better assay for Col11A1 as a biomarker for serous ovarian cancer. We do have a validated protocol for immuno-oncology checkpoint target profiling, and this slide is demonstrating the importance of tumor heterogeneity. These are the snapshot images showing expression profile of various IO targets in two different non-small cell lung carcinoma tumors. Clearly, there are differences in expression pattern of these IO markers between these two tumors. For example, LAG3 is showing expression in both tumor and stroma in first tumor. However, it is only expressed in stroma in the second tumor. And similarly, there are differences in the expression pattern of PDL1, TIM3, and PDL2 between these two tumors. One important observation is that TIM3 is expressed in almost mutually exclusive pattern here, excuse me, with PDL1, but these are co-expressed in the second tumor in some of the cells. And this co-expression analysis of immune checkpoint markers at the single cell level could potentially aid in understanding the resistance mechanism of checkpoint inhibitors, as well as it can bring insight into combinatorial therapies. One of the most widely used diagnostic applications for RNAscope is the detection of HPV in various tissue types, including head and neck cancers, cervical dysplasia, and cervical cancers. HPV status can be used in the clinical decision-making process to select patients for less aggressive non-surgical treatment. And we all know that P16 is a well-established surrogate marker for HPV status. However, P16 can be induced by other pathways which are independent of HPV. And this caveat led to interest in more specific assays for HPV detection. Among those assays are DNA and RNAscope in situ hybridization for HPV. And these assays have been evaluated, and there has been tremendous amount of work to evaluate and compare specificity and sensitivity of various assays for detection of HPV. As shown here, RNAscope is the highly sensitive and specific assay that can provide direct evidence of HPV activity. In this slide on the right side, I'm showing a Kaplan-Meier curve, which shows that the P16 positive but HPV negative patients, they have significantly reduced 5-year survival for oropharyngeal cancer as compared to the patients which were P16 positive and HPV positive. And this P16 positive but HPV negative was almost equivalent to P16 negative. Now, since P16 IHC may provide a false positive report for HPV status, reliance on P16 IHC is quite risky. On the other hand, HPV RNAscope testing is more specific for determination of HPV status, and it can provide favorable prognosis. So, for the development of RNAscope-based diagnostic tests, ACD has partnered with Leica Biosystems. This partnership leverages the combination of ACD's RNAscope in situ hybridization technology with Leica's Bond clinical advanced staining instruments. Leica has two types of automated histology systems. The first one is Bond RX and the second is Bond 3. These are basically identical systems. However, Bond RX is mainly used for research-grade tests such as biomarker validation, preclinical studies, assay design and feasibility, and prototype testing and development. On the other hand, Bond 3 is FDA-approved system, which is developed under design controls. And the assays performed on Bond 3 can be fully validated and registered with the FDA based on being performed on this FDA-approved instrument. The first clinical diagnostic test program that we worked on was with Merrimack Pharmaceuticals for their anti-ERB3 antibody therapeutic called MM121. They had determined that NRG1, which is also known as herigilin, was the biomarker that was correlating with patient response to treatment with MM121 in combination with standard chemotherapy. Herigilin is a secreted protein, so IHC was not an option, and they would not get enough material from a needle biopsy to perform QRT-PCR. Whereas it was enough for detection with RNAscope, and as we can see here, these dots are showing are the herigilin message that was detected in the small biopsy. And this is a negative control probe. This is a positive control probe, and the clear background with negative control and the good staining with positive control is giving us the confidence that the signal that we are seeing with herigilin is a real signal. And so finally, they partnered with Leica Biosystems and utilized RNAscope assay for combination diagnostic tests. For companion diagnostic test development and for the selection of patients in phase 2 clinical trial. This is the data from their phase 1 study, and this is showing progression-free survival in patients treated with MM121. It shows that breast cancer, ovarian cancer, and lung cancer patients with high herigilin responded much better when they were treated with MM121 in combination with standard of care therapy as compared to standard of care therapy alone. For example, here, these blue lines, these are the combination therapy with the patients having high herigilin level, and the black lines are the ones with the single chemotherapy, and it clearly shows better progression-free survival in these patients. This is a screenshot of a poster presented by Merrimack, and this is not a very clear poster, but the message that I want to send here is that they presented this poster with their academic collaborator at ASCO in 2019, and they reported that their results of the phase 2 clinical trial studies that that MM121 was used as a combination therapy, and they performed it on almost 500 patients, which came from 87 clinical sites on 3 different continents. And the performance of RNAscope was highly robust and reproducible on this large number of patients. Another example where RNAscope has proven to be most effective for biomarker assessment and development of diagnostic tests is for receptor tyrosine kinase FGFR1, 2, and 3, for which Bayer has a small molecule tyrosine kinase inhibitor. RNAscope was shown to be superior in terms of both specificity and sensitivity against IHC as well as nanostring assays. And originally in 2014, RNAscope was proven to be a better predictor of FGFR tyrosine kinase inhibitor sensitivity as compared to gene amplification. And it was also shown to be more specific than IHC. Based on these results, Bayer chose FGFR mRNA assay as a basis of enrollment of patients in their phase 1 clinical trial. And the positive results in that phase 1 clinical trial led them to partner with Leica Biosystems to have FGFR companion diagnostic assay development based on the RNAscope technology. RNAscope was then used for patient selection in a pivotal phase 2 and phase 3 clinical trials. Shown here is an example of a patient who was highly responsive, as you can see here between the baseline and the 11-week of treatment. This patient was highly responsive to the Bayer therapeutic tyrosine kinase inhibitor, and this was this was negative for FGFR amplification. However, the patient did show high-level expression of FGFR message in tumor biopsy, as you can see here. For further illustration, this is again a snapshot of ASCO poster describing the use of FGFR RNA expression as the basis of patient selection for phase 1 trial. And this trial involved 860 patients on which they ran FGFR1, 2, and 3 tests, again illustrating performance characteristics and high level of reproducibility and robustness of a clinical RNA in situ hybridization assay. This is a piece of data from Leap Therapeutics that they presented at Sept in 2019, and they showed that DKK1 expression correlated very well with a response to the treatment. And here, the top panel is showing the biopsy, patient biopsy, which showed high expression of DKK1 with H-score of 163. And the bottom panel is showing a tumor biopsy with low level of DKK1 with H-score of 7. And on the right side is the Kaplan-Meier curve, which shows the better survival of the patient with high DKK1 level as compared to the one with low DKK1 level. And therefore, they are going to use RNAscope as a patient selection test in their upcoming clinical trial, where patients will be selected based on having certain threshold of DKK1 expression. This is another example of use of RNAscope as a companion diagnostic. This is a press release from Roche's Ventana in 2013, where they described their intention for developing a companion diagnostic test for Insight using IHC assay for the small molecule IDO1 inhibitor. After a couple of years of development work, Insight decided to compare the specificity and sensitivity of other assays that became available. Almost 5 years later, in 2018, Insight presented this data in ASCO, showing the sensitivity and specificity of RNAscope platform in detection of IDO1 expression in tumor biopsies. And this was actually after a very short period of development work that we did, where we demonstrated that RNAscope is about 10 times more sensitive than IHC assay, that they spent several years to develop. And this is an example to re-emphasize that RNAscope is much more sensitive than IHC, and it has significantly faster timelines for development and verification in clinical context. I'm going to explain the experimental data in a little bit in my next slide. So basically, for that assay, Hela cells were stimulated with interferon gamma at a varying concentration for 18 hours and assayed for IDO1 expression by either RNAscope-ish or IDO1 IHC or a Western blot analysis. The plot on the right here is showing that IHC was 10 times less sensitive as compared to RNAscope or Western blot. And it also showed that there was almost perfect correlation between RNAscope and Western blot sensitivity. As you can see, the curve is overlapping. And so the strong correlation between RNAscope in situ hybridization and Western blot suggests a highly coordinated induction of IDO1 by interferon gamma at both mRNA and protein levels. Finally, Insight switched from IHC assay to RNAscope platform for their clinical trial. We performed a reproducibility study for IDO1. Excuse me. Shown here is the bar graph obtained from IDO1 RNAscope reproducibility testing in melanoma samples. And it can be seen here that the results from different instruments, as well as those obtained at different time points, are highly reproducible. And then they used RNAscope in a very large clinical study on over 2,500 patients. It was used in several large phase 3 studies. I'm showing this here to emphasize the fact that RNAscope performed consistently across many large studies involving multiple different tumor types. Unfortunately, the IDO1 did not show efficacy in improvement of the patients in terms of response to treatment, but the assay itself performed extremely well on a wide range of patients in clinical trial. And this IDO1 study addresses a frequently asked technical question about how RNA and protein correlate. It showed that RNA and protein correlate well when you have equally sensitive assay for detection of RNA and protein. Now switching gear a little bit. So RNAscope is equally relevant in CAR T therapy. And we know that CAR T therapy has revolutionized the immunotherapy world. So the therapeutic CAR T cells are generally administered systemically and must traffic to metastatic disease site and function within the tumor microenvironment. Since both are comprised of portions of various human proteins, it is almost impossible to distinguish engineered T cells from endogenous TILs using antibodies. But the RNAscope technology utilizes unique reasons for probe design and enables visualization of CAR T and PCR cell distribution in tumor microenvironment. While co-staining with a marker of activation such as granzyme B or interferon gamma enables assessment of activation state, co-staining with a cell type marker such as CD3 or CD4 or any other like CD4, like CD3, CD8, CD4, any other cell marker, it enables assessment of functionality and helps monitor anti-tumor activity. This is a biodistribution data from PCR T cell therapy in a patient clinical trial conducted by AdaptImmune. NY-ESO-1 is a tumor antigen expressed in many poor prognosis multiple myelomas. AdaptImmune developed a gene-modified autologous tumor-reactive PCR against the NY-ESO-1 peptide called SPEAR. They used RNAscope to understand mechanism of response and resistance to PCR T cells. they performed ISH on FFPE tumor biopsies prior to and following adoptive T-cell transfer in synovial sarcoma. And they showed a correlation between the anti-tumor effect and the infiltration of the engineered TCR T-cells into tumors. As you can see here, the trafficking of TCR T-cells is shown in red and CD3 shown in blue. This is data which was presented by Bellicum at ASCO GI recently in 2020. And using RNAscope in situ hybridization, they observed infiltration of TAR-positive cells into tumor tissues in all treatment samples, whereas endogenous PSCA expression and CD3 positive lymphocytes remained at the same level in baseline and on treatment samples. And Bellicum will be using RNAscope for their upcoming clinical trial. So in short, I would like to summarize about Pharma as a Services that we provide scientific pre-sales guidance to help you plan your study with one-on-one teleconference to understand your goals. Our scientists are available to present the summary data via WebEx or any other video conferencing platform to the key stakeholders. Our project manager will ensure timely delivery of your study and that objectives are met. These are some of the accolades that we have received from our happy clients. With this, we come to the end of this presentation, and we will continue to be online for a few more minutes. Please feel free to send your questions to our panelists who are waiting to answer your queries. Thank you.
Thank you very much, Alpana and Shauna. What an excellent presentation that was from both of you. We really appreciate it. We do have a couple of questions coming in. So before you leave today, please do, if you want to, please pop the questions in the question box and our team are online actively answering them right now. This has been recorded, and so in the next 24 hours, this presentation will be available online at our acdbio.com recorded webinars page, which you can download, which you can click and watch again for your enjoyment in case you missed anything. Do reach out to the PAS team and also the support team if you have any questions. If we don't get to answer your questions today, we will follow up via email in the next few days. With that, thank you very much.